Today we will be talking about a really important neurotransmitter in the body which is known as acetylcholine. Acetylcholine, or ACH, is a neurotransmitter, which is a chemical messenger that plays a vital role in transmitting nerve signals between nerve cells, or neurons, and from neurons to muscle cells. It is one of the most abundant and widely distributed neurotransmitters in the nervous system. Now, what are some super important functions of acetylcholine? The most important one is muscle contraction. One of the primary superficial functions of acetylcholine is its role in muscle contraction. In the peripheral nervous system, PNS, at the neuromuscular junction, acetylcholine is released from motor neurons and binds to receptors on muscle cells. This binding triggers a series of events that lead to muscle contraction, allowing us to perform voluntary movements like walking, riding, or lifting objects. Then autonomic nervous system control. Acetylcholine is involved in the autonomic nervous system, which regulates involuntary bodily functions such as heart rate, digestion, respiratory rate, and glandular secretions. In the parasympathetic division of the autonomic nervous system, acetylcholine acts as the primary neurotransmitter to slow down heart rate, promote digestion, relax smooth muscles, and stimulate glandular secretions. It also helps in learning and memory. Acetylcholine is also associated with cognitive functions, memory, and learning. It is involved in the processes that contribute to memory formation and retrieval. Studies have shown that changes in acetylcholine levels and activity in certain brain regions are related to memory and cognitive performance. We can also have attention and arousal with ACH. Acetylcholine is linked to attention and arousal mechanisms in the brain. It helps regulate wakefulness, alertness, and the ability to focus on tasks. Dysfunction in acetylcholine pathways has been implicated in disorders like attention deficit hyperactivity disorder, ADHD, and certain sleep disorders. One more function is pain transmission. In the spinal cord, acetylcholine has been shown to play a role in the transmission of pain signals. Modulation of acetylcholine levels in the spinal cord can affect pain perception and sensitivity. These superficial functions of acetylcholine represent some of its basic roles in the nervous system. However, it's important to note that acetylcholine's actions are highly complex and interconnected with other neurotransmitters and neural pathways, contributing to a wide range of physiological and behavioral processes in the body. Now, another question is, how this acetylcholine is made. The synthesis of acetylcholine involves a series of steps that take place within nerve cells, neurons, and require specific enzymes. Here is a detailed overview of the synthesis of acetylcholine. Step 1. Uptake of choline. Choline, an essential component for acetylcholine synthesis, is obtained from the diet through the intake of foods like eggs, liver, and certain vegetables. Choline is taken up into nerve cells from the extracellular fluid by a specific transport protein known as the high-affinity choline transporter, CHT. This transporter is responsible for moving choline from the extracellular space into the cytoplasm of the neuron. Step 2. Conversion of choline to acetylcholine. Once inside the neuron, choline undergoes a chemical reaction to be converted into acetylcholine. This reaction is catalyzed by an enzyme called choline acetyltransferase. Choline acetyltransferase is primarily found in the cytoplasm of cholinergic neurons, which are neurons that release acetylcholine as their primary neurotransmitter. Choline acetyltransferase transfers an acetyl group from acetyl coenzyme A, which is produced in the cell's energy-generating organelles called mitochondria, to choline. The result is the formation of acetylcholine and coenzyme A. Step 3. Packaging of acetylcholine into synaptic vesicles. After its synthesis, acetylcholine is rapidly transported into small, membrane-bound vesicles known as synaptic vesicles. This packaging is carried out by a vesicular acetylcholine transporter, VACHT, which transports acetylcholine from the cytoplasm into the vesicles. The presence of acetylcholine inside these vesicles allows for its storage and subsequent release upon nerve stimulation. Step 4. Release of acetylcholine. When an action potential, nerve impulse, reaches the nerve terminal, it depolarizes the cell membrane. This depolarization triggers the opening of voltage-gated calcium channels, leading to an influx of calcium ions into the nerve terminal. The increase in intracellular calcium concentration facilitates the fusion of synaptic vesicles with the presynaptic membrane, causing the release of acetylcholine into the synaptic cleft, which is the small gap between the presynaptic neuron and the postsynaptic neuron or muscle cell. Step 5. Binding of acetylcholine to receptors. Once released into the synaptic cleft, acetylcholine diffuses across the gap and binds to specific receptors located on the postsynaptic membrane of the target neuron or muscle cell. The binding of acetylcholine to its receptors initiates a series of chemical events that transmit the nerve signal to the target cell, leading to various physiological effects, such as muscle contraction or nerve signal propagation. Step 6. Termination of acetylcholine signaling. To prevent continuous stimulation, the action of acetylcholine is terminated shortly after its release. This is achieved through the action of an enzyme called acetylcholinesterase, ACE, which is present in the synaptic cleft. ACE rapidly breaks down acetylcholine into acetate and choline. The choline is then taken back into the presynaptic neuron through the choline transporter to be used in the synthesis of new acetylcholine, completing the recycling process. In short, the synthesis of acetylcholine involves the uptake of choline, its conversion to acetylcholine, packaging into synaptic vesicles, release into the synaptic cleft, binding to receptors, and finally, the termination of signaling through enzymatic breakdown. This intricate process ensures precise and controlled neurotransmission within the nervous system. We have discussed the synthesis, however. The release of acetylcholine is a crucial step in neurotransmission, allowing nerve signals to be transmitted from one neuron to another or from neurons to muscle cells. This process occurs at specialized points of contact called synapses. Let us get to know about how acetylcholine is released. First, nerve signal or action potential. The process of acetylcholine release begins when an electrical signal, known as an action potential, travels down the length of the presynaptic neuron. This action potential is generated when a neuron receives input from other neurons or sensory stimuli, and it serves as a means of transmitting information along the nerve cell. Then depolarization of the nerve terminal. As the action potential reaches the nerve terminal, axon terminal or presynaptic terminal, it causes a rapid depolarization of the membrane. This depolarization leads to the opening of voltage-gated calcium channels in the presynaptic terminal. Next comes the influx of calcium ions. The opening of the voltage-gated calcium channels allows calcium ions to flow into the nerve terminal from the extracellular fluid. This influx of calcium ions is a crucial event in acetylcholine release. Then vesicle docking and fusion. Within the presynaptic terminal, there are small, membrane-bound vesicles called synaptic vesicles that contain stored acetylcholine. The increase in intracellular calcium concentration triggers the docking and fusion of these synaptic vesicles with the presynaptic membrane. After that, exocytosis of acetylcholine. As the synaptic vesicles fuse with the presynaptic membrane, they release their contents, which include acetylcholine, into the synaptic cleft. The synaptic cleft is the narrow gap between the presynaptic neuron and the postsynaptic neuron or muscle cell. Then, diffusion and binding. Released acetylcholine molecules now diffuse across the synaptic cleft and bind to specific receptors on the postsynaptic membrane. These receptors are proteins that are sensitive to acetylcholine and initiate the transmission of the nerve signal to the postsynaptic neuron or muscle cell. And lastly, termination of signaling. To prevent continuous stimulation, the action of acetylcholine is rapidly terminated in the synaptic cleft. This is achieved by the action of the enzyme acetylcholinesterase, which breaks down acetylcholine into acetate and choline. The choline is taken back into the presynaptic neuron to be used in the synthesis of new acetylcholine, completing the recycling process. Overall, the release of acetylcholine is a precisely regulated process that ensures proper communication between nerve cells and muscle cells, enabling the coordination of various physiological and behavioral responses. That was all for today. Keep watching for more.
In the race against time, there is a game changer. Welcome to Medical Learning Reimagined, a world where over 40 specially curated courses are just a tap away. Dive into the mysteries of genetics, or unravel the complexities of cell physiology. Why stick to the old ways when you can learn smart? Harness the power of digital education. Leap into the future, mastering medicine one swipe at a time. sqadia.com. Medicine modernized.